Okay, well, let's go ahead and get started. To begin with, we're going to review mitosis. So we have two cell division processes. What are they? Mitosis and meiosis, exactly. And what's happening in both of them? What is the purpose of mitosis?
Cell division.
Who said it first? Us all? Oh, sorry, Chris, I forgot to put this on.
Ladies always. They always.
Yes. Okay. Or you need cells for the body so your body can grow bigger and so you have enough cells to make all the different parts that you need. So mitosis, the purpose of it is for growth. And if you damage those cells or they wear out, you need to be able to replace them, repair. In other words, you start out as a fertilized egg. Is that egg diploid or haploid?
For us, it's diploid.
It's diploid. For most organisms, it's diploid. Okay, there are some organisms that are other ploides, but animals typically are diploid. I don't know of any that aren't. Okay, so we start with a diploid somatic body cell, and it divides by meiosis. How many times does it divide? By mitosis? Just once. And in the process, it separates. First, it makes a copy of all the DNA. When?
In S phase.
During S phase. And then, so here's our cell cycle. What's the longest phase, typically? G1. If you leave G1, then you go into S phase, and what do? Replicate the DNA by a process called DNA replication. Then you go into G2, where you're just kind of waiting to be ready to finish dividing. And then you go into M phase when you actually divide. And so, if a cell divides, what has it done before it actually divides? Has replicated its DNA, so you have two copies of the DNA. And what are you doing in mitosis? You're separating those two copies of DNA from each other, and then once you do, the cell then pinches in half by what process? Mitosis is the division of the DNA. Cytokinesis is the division of the rest of the cell. Okay? So you divide in half, and now you've got two cells that are still both 2N, or you're getting more cells of the same type. So you can grow bigger. You haven't changed anything. What are we doing in meiosis?
Creating sex gametes.
Yeah, the only purpose of meiosis is sexual reproduction. All right? And by sexual, we mean we are mixing up the DNA from two different organisms to create a new organism that's similar to the parents, but not exactly the same. Why is that important? Why do we want variation?
That is evolution.
Yes, that enables evolution. It gives something for natural selection to work on, to select for or select against. If everybody was the same, we'd either all survive or we'd all die. But you wouldn't have some surviving in some environments and others not. So there would be no ability to adapt. So sexual reproduction, meaning the purpose of meiosis is to make what?
Gametes.
Gametes. Exactly. And gametes have to be, by definition, half of the normal ploidy. In animals, the normal ploidy is diploid, 2N. What's half of 2N? 1N. So the haploid state would be 1N. If your normal state is hexaploid, you have 6 of every chromosome, what would your haploid state be?
3N.
It'd be 3N, exactly. Why do you want to make haploid gametes?
Because you don't want extra chromosomes in the final product?
Exactly. The purpose of gametes is to unite in a process called fertilization, and that makes one cell, your zygote, which is now diploid again, and then what does it do? Divides by mitosis to make more cells to grow your body. All right? How do we make haploid gametes? We have to divide not once, but how many times? Twice. And at the end of the first division, am I haploid or diploid? I'm already haploid, but my chromosomes are still replicated. So here I have replicated homologous chromosomes. Here, each cell still has replicated chromosomes, but I only have one of each different kind of chromosome. And then what happens in the second division? You separate, what are these? Sister chromatids. And I end up with 4 haploid cells, each of which has one copy of each different kind of chromosome. All right. Why is this not showing up there? Try that again. Okay, I wanted to show you a movie of mitosis. Draw out on the back of your quiz sheet, draw out the steps, the phases of mitosis. Prophase / prometaphase, then anaphase, telophase, metaphase, anaphase, telophase. Draw it for a cell where that is 2n equals 4.
You got to draw the cell for each phase. What's going on in there? For mitosis. And the cell's 2n. No, she has to draw the back name. She's actually asking. Oh, yeah. You said meiosis? Mitosis. Mike?
Well, I don't know why we're not having anything work today. Printer wouldn't work. Projector's not working. So we'll try it again. Okay, 2n equals 4 means you have a total of how many chromosomes? 4. How many different kinds of chromosomes? What's n equal to? N is equal to 2. What does your DNA look like when you're still in interphase? Can you see individual chromosomes?
It's basically a mess.
Yeah, is the DNA fully condensed or partially condensed?
He wants to do T1 S G2 of that. Don't just do the mitosis. Just the M type of something? Yeah. Because the S phase is like hardly anything. Let's see if we can really tell the M.
Okay, well now we've got absolutely nothing happening. Great. Okay, I feel like it's making an effort to try to work at least. So this comes here, that is correct. Ha! Success. All right, here is a cell that's about to divide. This happens to be a plant cell. It is in what phase of the cell cycle? It's just coming out of interphase. We know it's not in interphase right now because what can you start to see?
Starting to condense a little bit.
Yeah, we still have a nuclear envelope that's separating it from the rest of the cytoplasm, but what can we see forming inside there? Does it just look randomly something in there, or are we starting to see actual structures? So my chromosomes are starting to condense. So I'm not in interphase anymore. I'm in what phase? Prophase. Because what happens in prophase? Your chromosomes start condensing. Why? What got turned on at the G2M checkpoint? MPF. And what is MPF? Yes, good job. MPF stands for maturation promoting factor. What molecules is it made out of? It's a CDK plus what? Plus cyclin, exactly.
Yeah.
See if we can get some markers that work. Okay, so it's a CDK. We're just going to call it M-CDK because it's the CDK that takes you into M phase. And what has to be bound to it? Cyclin. I think it's cyclin B, but we're going to call it M-cyclin because it's the cyclin that needs to bind to this CDK. What else has to happen to this CDK to be able to be activated? All during G2, the cell is making M-cyclin. You already had M-CDK in the cell. Here's my CDK. This is its allosteric site. What binds here? What binds here? It is a kinase. The CDK is a kinase. Cyclin binds here. And we're talking about M-CDK. So which cyclin binds here? M-cyclin. Is that sufficient to turn the CDK on? Yeah, what else has to happen? During G2, it has been phosphorylated in two sites. One is the activating site. One is the inhibitory site. So all during G2, cyclin's been binding to CDK, but we're not on yet. What has to happen before you actually activate it? Boom, all at once, right here at the end of G2.
You have to pick off the inhibitor site, right?
Yeah, you have to get rid of that phosphate. How do you remove a phosphate? What enzymes do that? A phosphatase. And what's the name of the phosphatase that gets turned on at the end of G2 if the cell is ready to divide? It's called CDC25. Okay? So CDC25 is a phosphatase, and it removes that phosphate, which turns the kinase on. And now when my kinase is on, when my CDK is on, what does it do? What binds in its active site? Now, I don't mean it binds directly in the active site, but I mean this kinase will phosphorylate things that will eventually cause this to happen.
It's going to start condensing, and then it's going to
Here we're seeing our chromosomes condense. Why? Because this kinase caused what to get phosphorylated? Condensins. What are condensins?
They pull along the fiber of the DNA in order for it to...
Exactly. They are motor proteins that bind to what? DNA. And they cinch up the DNA and cause it to get a bunch more loops in it. That makes it shorter and fatter, or more condensed until it gets chunky enough that we can see it. Okay? So what's happening in prophase? My chromosomes are condensing. Why did I leave G2 and I'm now in prophase? Because my chromosomes are condensing. That doesn't happen in G2. That's a definition of being in M phase. So what are the phases of M phase? What's the first phase? Prophase, then prometaphase. And we're just going to call them one phase because the same events are happening. They're just starting in prophase and they're finished in prometaphase. Different species. Like another thing that's going to happen is your nucleus is going to disappear. Some species, it disappears in prophase. Other species, it disappears in prometaphase. So I'm not going to separate them out into different phases. So what are the three key events that happen in prophase, prometaphase?
So migration of our centromeres.
Not centromeres.
Centrosomes.
Centrosomes, which are what?
There's too many of those central whatever.
They are,
There's
yep.
like 3 of them, and I can't keep track of them all.
So centrosomes, some means body. It's a body that does what?
It lays down the motor fibers to the...
It builds microtubules.
Yeah,
It's a microtubule organizing center, an MTOC. All right? And you're absolutely right. It's building the fibers that are part of the mitotic spindle. So I'd like you guys to be drawing this out, but I guess I'll draw it up here too. Okay? So in prophase, what's happening? My chromosomes are starting to condense. What do they look like when they appear? They look like that. But how are we going to draw them? Like this. Meaning, have they been through S phase? Yes. What are these? Sister chromatids. If I drew this cell, and that was all the chromosomes in the cell, is this cell haploid or diploid? It is haploid. And how would you describe it? It's 1n equals 3. Or how many different kinds of chromosomes are there? 3. N is equal to 3. And how many of each kind do I have? 1 of each kind. That's why I'm haploid. Okay? What if when I condense I have 2 of each of these kinds of chromosomes? Then I am 2n equals 6. All right? So we'll do a cell that's 2n equals 6. So that's the first thing that's happening. My chromosomes condense. Why? Because MPF phosphorylated what? Condensins. What else is happening? What's disappearing? My nucleus is disappearing. Why? Because MPF phosphorylated what? What makes your nuclear structure stable? What are the fibers that are underlying the nuclear membrane and giving it its shape? Not lamina, lamins. Nuclear lamins. Lamina is going to be something totally different. It's your basement membrane under epithelial cells. Okay? So what else gets phosphorylated, or what else binds in the active site of this CDK? Nuclear what? Lamins. So condensins get phosphorylated, nuclear lamins get phosphorylated, and that causes the lamins to disassociate from each other. What kind of fibers are nuclear lamins? Intermediate filaments. And remember how intermediate filaments form? I'm not going to go through that all now, but you had the 2 dimers with the coiled coils that twist around each other. They're directional. And then 2 dimers twist around each other in opposite orientation. So I now have a tetramer that's not directional. And then those tetramers bind end to end to make big, long fibers. Yeah, when I phosphorylate them, they don't bind end to end anymore, and that breaks down the nucleus. All right, what else is happening? Chris said it. These regions that are producing fibers, what are these fibers called? Kinetochore microtubules, because they're connecting to what proteins right here at the centromere? This is the centrosome. It's forming microtubules that attach to proteins located at the centromere. And what are those proteins called? These proteins is a big complex of lots of proteins, but they're called kinetochore. Kinetochore proteins. So what do we call these fibers?
Kinetochore fibers.
Yeah. They're microtubules because they're made by a microtubule organizing center, but they are called kinetochore microtubules. All right? What other kind of fibers am I producing here? My centrosome is also producing fibers that just reach out and interact with each other. They don't interact with the DNA. What do you call those fibers? Yep. So they don't bind to the kinetochore protein, so what are they called?
Oh, there we go.
Non-kinetochore microtubules. Does that make sense? So my centrosomes are making 3 different kinds of fibers. Kinetochore microtubules, which attach to the chromosomes. Non-kinetochore microtubules, which interact with each other from opposite sides. And they also have fibers coming out the back, which are called aster fibers. And these attach to proteins in the cell membrane and anchor you. Why is that important? Because what is the function of these fibers? They have motor proteins that are interacting with the kinetochore proteins, and they're causing tension on the chromosome pulling this way. But what else is attached here? In mitosis, the centrosome on this side forms microtubules that attach here, but the centrosome on this side also forms one. So do I have an equal force pulling on these chromosomes in both directions? So what's going to happen to the chromosomes if they have fibers attached to them on both sides? You're playing tug-of-war with exactly equal-sized teams. The chromosomes are like the flag on your rope. They're all going to end up where? In the center. And what do we call the center of the cell? The metaphase plates. They're all going to end up there. So in prophase, my chromosomes are condensing, my spindle fibers are starting to form, and my centrosomes are starting to migrate. By prometaphase, my centrosomes are pretty much done migrating. They have attached to all the chromosomes. And how have they attached? I have at least one kinetochore microtubule coming from just one side or coming from both sides?
Both sides.
Coming from both sides. And there's tension on these microtubules because of the motor protein interaction, and also because the microtubules are being built and also shortened. We have dynamic instability going on, so that's also causing the tension. And the tension causes my chromosomes to all line up. And as soon as they're all lined up, I am no longer in, I'm going to get rid of one of these sets of chromosomes because it's too messy with so many. Well, I guess we'll just have them all. Okay. Okay, so I have 6 chromosomes. How are they lined up?
They're stacked.
End to end is the way we say it. Now, different kinds of chromosomes will always line up end to end. But how are homologous chromosomes lined up right now? With relationship to each other, does where this homologous chromosome is have anything to do with where its homolog is? No. They're lined up totally randomly, and all the chromosomes are end to end along the what? What's this called? This imaginary line that's dividing the cell? The metaphase plate. All right? What lined them up? What caused this to happen? The kinetochore microtubules playing tug-of-war because there's tension going this way and going this way. How come the sister chromatids don't get pulled apart if I'm pulling on the kinetochore on this side and the kinetochore on this side?
They're bound together right now.
Yeah, what's binding them together? We have proteins, once again, motor proteins, that are holding them together. What are those called? Cohesins. And they've been holding the sister chromatids together ever since S phase when we first made sister chromatids. So they're keeping them organized. What other fibers do we have? The black ones are kinetochore microtubules. The blue ones don't interact with the chromosomes. They come out and interact with each other. And then there's motor proteins that walk along one and push against the other one. And that's going to, they push against each other, and that's going to expand my cell. So what's the next phase? The next phase is, I didn't draw that very well, is anaphase. And now my cell is longer and skinnier, or it's changing shape because of this spindle apparatus. What is the spindle apparatus? It is the 2 centrosomes and the 3 kinds of fibers that are interacting. All right? In anaphase, what happens? How do I know I'm not in metaphase anymore? Yeah, your DNA is now separating. What are we separating in mitosis? Sister chromatids or homologous chromosomes? Sister chromatids, and that's why we're going to end up still diploid at the end, because we're getting a copy of every chromosome going into the daughter cells. So this is metaphase. What happens in metaphase? There's prophase, prometaphase. Now we're in metaphase, then anaphase, then telophase, and then cytokinesis. So what happens in metaphase? Do we have a checkpoint going on there? Who gets turned on at this checkpoint? APC, anaphase promoting complex, or the thing that makes anaphase happen. What is APC? APC is an E3 ligase. What's that? E3 ligase is an enzyme that's the final step in adding what to proteins that you want to destroy by the proteasome? It's found in every cell. It is ubiquitous. What's it called? Ubiquitin. And the process of adding ubiquitin to a protein is called ubiquitinating or ubiquitolating. I can never remember. Can't say it. Okay? So what does APC do? We talk about APC destroys things, but APC doesn't actually destroy anything. What does APC do? It tags it with ubiquitin, and when you're tagged with a series of ubiquitins, who do you get attracted to? The proteasome, and the proteasome then destroys you. So what are some targets of APC? Let's back up a little bit. What's the question we're asking at the M phase checkpoint? Are my chromosomes lined up properly? The way the cell determines that is it actually measures the tension that's on the chromosome. And if you have equal tension in both directions, then it figures your chromosomes are lined up correctly. All right? So if the answer is yes, then it's okay to go into anaphase. How do I get the cell to go into anaphase? What gets turned on? APC, which is an E3 ligase, and it adds ubiquitin to what? What's holding my sister chromatids together so that they won't break down? Cohesins. So what's the path here? Cohesins. I'm not sure what the best way to write this is. They're currently on when APC is off. Okay? So APC off, APC on. If APC is off, my cohesins are on, and can I pull my sister chromatids to get apart? No, because the cohesins are holding them together. Well, what regulates this? The thing that when APC gets turned on, then a protein called, let's see, when APC is off, securin is bound to, securin is inhibiting separase. So I need an eraser. When you draw a line like this, it means you're causing something to happen. When you draw a line like this, it means you're preventing something from happening. You're inhibiting it. So normally, there is a protein in the cell called securin. And when securin is on, it is inhibiting separase. So separase is off. When APC gets turned on, securin gets ubiquitinated, and what happens to it? It gets destroyed by who? The proteasome. Can you see that? Proteasome. When securin is off, it's no longer keeping separase off, so separase is now what? Separase is now on, and what separase does is it inactivates the cohesins. So we don't destroy the cohesins, we just turn them off. They're proteins that can reach out and grab things, or they can let go of the things and just sit there waiting to be used later. And then when they're on, they reach out and grab the DNA. When they're off, they're just sitting here like this. All right? So it inactivates who? The cohesins. And now are the cohesins holding the sister chromatids together anymore? No. And now what happens? How do I know I'm in anaphase? Because I now have space between whatever DNA I'm separating. All right? Let's see. I'm not drawing this very well. Where are they located? I don't like having 6 chromosomes. This is too hard. Okay? Why do I now have space between my sister chromatids?
Because the existing fibers are still providing tension.
Exactly.
When the cohesive release to allow them to pull apart.
So why is this called anaphase promoting complex? Because the definition of anaphase means your DNA is separating. What was preventing my DNA from separating in prophase and metaphase? Cohesins. And so what happens at the M phase checkpoint? We turn off what? Cohesins. Who turns it off?
APC.
MPF, but, no, not MPF, APC, but this happens directly or indirectly?
It's
Indirectly. What does APC actually turn off? APC adds what to what? Ubiquitin to securin. Now securin is no longer inhibiting who? Separase, and separase does what?
This would be a heck of a lot easier to remember if we just changed the names to the Game of Thrones people and how they killed each other. This would be so much easier to remember.
And it binds, yeah, I haven't seen it either. And it inactivates the cohesins, okay? So the key event that takes you, it's called anaphase promoting complex because ultimately it inactivates the cohesins. If you don't have cohesins holding your DNA together, now you can separate the sisters. All right? And as soon as you're separating, you're no longer in metaphase, you're now in anaphase. What else is happening? My spindle fibers are doing what? They're getting shorter, and my motor proteins are walking along them and pulling too. Okay? So this is anaphase. Anaphase happens pretty fast. The DNA is getting separated to each side.
Can I just ask one question on those motor proteins? We learned that we had like dyneins, and what was the other one? Is it the same motor proteins as the previous one?
Yes, it is. And I think it's a special kinesin.
Okay.
Well, no, let's see. I think it's a special dynein, actually. Because dyneins usually walk towards the center of the cell, but in this case, they're walking towards the outside.
Okay.
Although they're always walking towards the centrosome. And so, I can't remember. Somebody needs to look that up.
I was looking at this again, I was like, I'm kind of interested.
Yeah, I know it's a special form of a motor protein that's walking opposite of the way that they usually walk.
Okay, thank you.
And they're involved in here, but I can't remember if it's dyneins or kinesins. But you know it's one of them because we're walking along what? Microtubules.
Yeah.
All right? What else begins to happen usually in anaphase? Cytokinesis is usually beginning to happen at this same time. They don't have to be coinciding, but normally they are. And then the last phase is what? Telophase. And what happens in telophase? The exact opposite of prophase, prometaphase. So what three things are going to happen? My chromosomes are going to decondense back into what? Chromatin. My nucleus is going to reform, and my centrosomes are not migrating, and they're not making the spindle fibers anymore. They're going back to being normal centrosomes that are next to the nucleus that are now making microtubules that you need to pull your ER out and localize it, and pull your Golgi in and localize it, and make roadways to transport vesicles from place to place. Or my big box of Legos, my tubulin molecules that I can build microtubules out of, is no longer making a spindle apparatus. It's now making what? The normal microtubules that you normally have in a cell. And I'm no longer in M phase, because the classic thing of M phase is you have a spindle apparatus. Now I'm in what? G phase. I have decondensed chromatin, I have a nucleus, I have one centrosome, and I have normal organelle structure. Okay? What else usually finishes happening along with telophase? Cytokinesis. Cytokinesis. So, if I was to draw this out with the chromosomes still condensed so we can see what they look like, what would my cell look like? Would it be haploid or diploid? It'd still be diploid. How come? Because did I get one big chromosome and another big chromosome? Or here we see two big chromosomes going to this side, two going to this side. Two skinny ones going to this side, two to this side. Two wiggly ones to this side, two to this side. Or do I still have two of every chromosome?
You just have as much total of DNA.
I just have one molecule of DNA per chromosome now because I'm in what phase? G1. And if I go into S phase, then I'll replicate again. Okay? So that's all the stuff you learned last time. Okay, here's our movie. You saw this last time because I found it on the internet. But
Differential interference contrast microscopy is used here to visualize mitotic events in a lung cell grown tissue culture. Individual chromosomes become visible as the replicated chromatin starts to condense. The two chromatids in each chromosome remain paired as the chromosomes become aligned on the metaphase plate.
okay, what phase are we in now?
The chromatids then separate and get pulled by the mitotic
There's
two
anaphase.
daughter cells. The chromatin decondenses as the two new nuclei form, and cytokinesis continues to constrict the remaining cytoplasmic bridge until the two daughter cells become
And now we're in telophase. And now my cells flatten out. They reattach their junctions, like Chris was saying, and become normal cells back in G1 again. Okay? All right. So here are just slides saying what's going on in each of these stages. We just went through all of these. But here I am in interphase. My DNA is in what structure? Chromatin. I have how many centrosomes? 2. You duplicated them during interphase. But they are making normal microtubule structure, interphase structures, not a mitotic spindle. Typically, interphase, in the typical life cycle of a cell that divides once a day, you can assume interphase is about 22 hours, and dividing takes about 2 hours. That's kind of standard for a mammalian cell. Once it decides to divide, it'll take about 2 hours. How long interphase is totally depends on what kind of cell you are. If you're a liver cell, it's about a year before you divide. If you're a brain cell, you're never going to divide again. If you're a gut cell, you're going to divide twice a day. All right? But when you actually divide, how long does it take you? About 2 hours. Does it matter if I'm a liver cell or if I'm a gut cell? No, because they both have the same amount of DNA, and the process takes a certain amount of time. Okay, actually I'm thinking of S phase. S phase would be the same in all of them. How long this takes does depend on what kind of cell you are. If you're an ovary cell making eggs, it might take you 50 years to finish that division. Okay? All right. So the cell cycle is divided into 2 phases, interphase and M phase. Today we're focusing on what happens in M phase. We've talked about M phase being regulated by MPF. And what are the 3 things that MPF does in prophase? Phosphorylates lamin, which causes what? Nucleus to disappear. It causes chromosomes to condense by phosphorylating condensin motor proteins. Then, how do I leave M phase? We just talked about how at the M phase checkpoint, we activate APC. That destroys securin, which inactivates cohesins, and lets me go into what? Anaphase. Another target of APC is M-cyclin. So what else happens when APC is off? M-cyclin is on, or it's working. It's bound to your CDK and your MPF is on. When APC gets turned on, what does it do? M-cyclin gets what? Ubiquitinated, and who destroys it? The proteasome. Now, I don't see the effects immediately, but if I no longer have M-cyclin phosphorylating my condensins, what's going to happen to my DNA? Yeah, as the condensins become dephosphorylated, which will happen over time because there's always phosphatases present in cell. They're just acting slowly, so eventually they'll catch up. My condensins will eventually become dephosphorylated, and then what will my chromosomes do? Decondense. But it takes a little bit of time till you see that effect. It doesn't happen in anaphase, but it is happening by the time we're in telophase. What else happens in telophase? My nuclear membrane is reforming. Why?
Because we're,
M-cyclin got ubiquitinated and destroyed, so MPF is off. If MPF is off, am I phosphorylating lamins anymore? No. And when they're not phosphorylated, remember, all your cytoskeletal filaments are self-assembling. If they're not phosphorylated, what will they automatically do? Recombine and make your nuclear lamina, and that reforms your nucleus. What else happens when MPF is off? Your centrosomes stop migrating and stop having greater dynamic instability and making a mitotic spindle, and they just go back to the normal activity level. Okay? So everything that happened in prophase is reversed because we turned MPF off in metaphase when we ubiquitinated who? Cyclin. When you do the last lab, which is the last day of class, we're going to just look at dividing cells. It's a very relaxing lab. And you're going to identify cells in different stages. And then what you're doing in your lab report is just preparing you for the test, because you are going to be writing out what am I seeing in each of these stages, and you're also going to be explaining what's happening molecularly that I can't see, but it's causing the things that I can see to happen.
So the checkpoints and the on and off switches and what's going on.
Exactly. Okay? All right. So that's mentioning all those key events that we just went over. Vocab check. What's the difference between a cohesin and a condensin?
Cohesin are the proteins that hold together the chromosomes.
Yes.
Condensins pull them into the coil structure.
They're both motor proteins, but they're doing different jobs. Cohesins are just holding what together? Sister chromatids. And condensins are doing what?
Coiling them up.
An individual molecule of DNA, it's ratcheting it up and making coils to make it shorter and more condensed. Okay? And they both don't get destroyed, they just get turned on or get turned off at different phases of the cell cycle. All right. Another vocabulary check. Centromere, centrosome, centriole. Okay, do you know the difference between all of those?
Structure that lays the microtubules out, and the centrioles, centrioles are the individual two structures inside there.
Yep.
To do that.
Only in animal cells.
Yeah, and then centromere is not the center of the.
It's the more highly condensed region of the chromosome. It's the pinched-in region, usually in the middle, but it doesn't have to be. And what proteins are located there?
Clear,
Kinetochore proteins.
yeah.
Okay, here's prophase. How do you know? Because my DNA is what? Appearing.
And it starts still at the nucleus.
Why? Because MCTK triggered what? Condensation of chromosomes, and we're starting to build what? You can't see it, but the spindle apparatus. They're too small to see, but they're there. Here is a what? Replicated chromosome. This is the centromere. Do my chromosomes look like this in G2?
No.
Yes and no. They do in that they have two sister chromatids. They don't in that they're not fully condensed like this. You're only fully condensed like this when?
Prophase.
In M phase. What caused you to be fully condensed like this? MPF turned on what? Condensins. Okay? So we've talked about all of that. Condensins and cohesins. So here's a picture kind of the cohesins. They're just reaching around, grabbing and holding the DNA together. They're lining the whole length of the chromosome. What do condensins do? Condensins take this DNA and they ratchet it out into loops. And that makes my DNA that was this long now only be this long, but a lot wider. Okay? All right. We've talked about all these things before. I'm not going to go over that again. There's a diagram of a cell. How would you describe that cell?
That's metaphase.
That would be metaphase. And it looks to me like a 1n equals 2 cell. Has two different kinds of chromosomes in it and just one of each one. Here we are. So, what caused me to line up in metaphase? Which fibers? The kinetochore microtubules. Kinetochore microtubules have two jobs during M phase. The first job is to attach to the chromosomes, play tug-of-war, and get them to do what? Line up. The second job is, once we destroy cohesins, it is to separate them and take you into anaphase. So same microtubules, two functions. It all depends on whether cohesins are on or whether cohesins are off. How do you turn cohesins off? Yeah, you activate APC, which happens when? In the M phase checkpoint, if what? What's the question the cell asks?
If all the chromosomes are lined up.
Yeah, is my DNA lined up properly? Okay, which it determines by measuring tension. All right. How else is the cytoskeleton involved in M phase? Once I've finished separating my DNA to both sides of the cell, now I divide the rest of the cell in a process called cytokinesis. Does this depend on microtubules or on actin? It depends on actin microfilaments. Where is my actin normally in a resting cell that's in interphase? It's all underlying the whole membrane everywhere, and a lot of it is involved in contractile actin fibers attached to focal adhesions that are attaching my cell, anchoring it to the substratum. So cells are normally flat and attached to things. When you go into M phase, they let go of what they're attached to. They round up. They look totally different because I don't have actin giving them their normal shape, because all my actin pile of Legos is being used to make what? A contractile actin ring. All right. Prometaphase, we're now phosphorylating what? The nuclear lamins, and what disappeared? I no longer have a nuclear membrane. I still have the centrosomes, and I'm still lining up the chromosomes. We've talked about nuclear disassembly before. Now I'm in metaphase. How come everybody's lined up along the metaphase plate? Who's doing this? Spindle apparatus, which is the 2 centrosomes with the 3 kinds of spindle fibers that they're making. Kinetochore, non-kinetochore, and the other one. Now we're in what phase?
Anaphase.
Anaphase. What's getting separated? If it's mitosis, sister chromatids. If this was meiosis, what would be getting separated? The big difference between mitosis and meiosis is how the chromosomes line up in prophase. In mitosis, the goal is to end up with cells that are different or the same as what you started with. The same. So how do I line up? End to end. So a copy of everything goes to both sides of the cell. In meiosis, the goal is to do what? Reduce my ploidy, my number of chromosomes. Instead of having 2 of every chromosome, I only want 1 of every chromosome. So how are my chromosomes going to line up? Side by side. What's lining up side by side? Chromosome 1 and chromosome 7?
They have to exactly match up.
Yeah, the homologous chromosomes. 2 chromosome 1s line up next to each other, 2 chromosome 7s line up next to each other, and then they get segregated to opposite sides. All right, here I am in telophase. It's just the reverse of everything else forms. So you should be able to say, why does the nucleus reform? Why are my chromosomes decondensing? Et cetera, et cetera. And then in cytokinesis, we now divide the cytoplasm. This is an animal cell. How is the cytoplasm getting divided? What's pulling the membrane in here and here? It's the contractile actin ring. And it is interacting with the microtubule fibers too. They are actually both dependent on each other, but they're working together. So we've already watched the movie of cell division in plant cells. Or actually, the one we watched before was in animal cells. This one's in plant cells. What's different about in plant cells?
Cell wall.
At the very end, it's how cytokinesis happens. I'm not going to watch this movie again. So here's kind of what you're going to see in our lab. You'll see cells. They're plant cells, so they have cell walls around them, and they're in different stages. What stage is this one in? Yeah, it's in interphase. You don't know if it's G1 or G2.
okay.
But you know it's in interphase because can I see individual chromosomes? Nope. This is all interphase. And do I have nucleoluses? And so the nucleolus disappears also when the nucleus disappears. What phase is this? Metaphase. What phase is this? I'd say anaphase there. What about this? Could be prophase or it could be telophase. It's kind of hard to tell, but it looks to me like that's one cell. It's just really giant. So, yeah, these are probably prophase. My chromosomes are condensing, but you can't really see them individually yet. Where's a good telophase cell?
Wouldn't the one above it that you just pointed at, that would be prophase where it's smaller, and those are elongated cells.
This one?
Well, down. That one there's quite large. That looks like more like it would be a telophase. It's going to separate the 2 large.
Yeah, it could be early telophase because my cell is really elongated here.
Up and to the left, or to the left, that one looks like it would be.
Yep.
Would be prophase, the smaller cell starting to condense.
Exactly.
Okay,
So it's kind of a judgment call, and you'll get to make your own decision. What about this one? That looks to me like that's telophase, and it's right near the end because I've got 2 cells, but you can still kind of see the chromosomes, but the nucleus is reforming, and you can see a new cell plate forming between them. How do animal cells do cytokinesis? By forming a contractile actin ring that pinches the membrane in. How do plant cells do it?
They lay down a new wall.
Yeah, they lay down a new wall. Or basically, here's my cell. I've divided the DNA to both sides. I've reformed my ER and my Golgi and all that stuff, and I now package up cell wall material into vesicles, and I line those vesicles up along the middle of the cell. And when you have enough of them, they can then fuse with each other and fuse with this membrane, and what do I end up with? two cell membranes, a continuous cell membrane with cell wall material in between. That's called making a new cell plate. So plants do cytokinesis by forming a what? Cell plate. Animals do cytokinesis by doing what? Contractile actin ring. Good. Okay. So here's our spindle apparatus. Consists of two centrosomes, aster fibers. What kind of fibers are these? Kinetochore microtubule fibers. Their first job is to line us up in what phase are we in here? Metaphase. Your second job is to separate the sister chromatids if it's mitosis, or the homologous chromosomes if it's meiosis 1. Okay? All right. Before you can do M phase, what are two things that have to happen? Before you can divide, you need a copy of all your DNA. When did you do that? S phase. So clear back here. Long time ago, right? And what else do you need? Normally cells have one centrosome. You need two centrosomes. So that also duplicates during interphase. All right. You've learned about dynamic instability and how forming microtubules is kind of like the fisherman casting out. And if it anchors to something, then your microtubule gets stabilized. If it doesn't, then it doesn't. The rate of dynamic instability increases about 10 times, an order of magnitude, during mitosis. So you're forming microtubules a lot faster than you were before and reeling them in again a lot faster. And what's the primary protein that's stabilizing my microtubules?
This is backwards.
Every time the spindle fiber goes out and hits a chromosome, what's it interacting with that stabilizes it? The kinetochore proteins, and that stabilizes it. Yeasts just have one spindle fiber coming in on either side. Humans have like 40 on either side. So different species have different number, but you need to have the same amount anchored on both sides so you have equal tension on both sides. That causes you to line up along the metaphase plate. Some of these microtubules don't reach out and interact with kinetochore proteins. Instead, they reach out and interact with each other, and that stabilizes them. And I now have motor proteins here, and what does that do? It causes these two to get pushed in opposite directions, and that elongates my cell. Why is it better? Is it better to be round when I'm dividing my DNA, or oblong? Oblong. And how do I become oblong? What kind of fibers are doing that? The non-kinetochore microtubules are doing that. Okay? So spindle fibers involved in all of that. So what's the centrosome cycle? During interphase, do I have more or less dynamic instability? Less during interphase. During M phase, I have more. And then that ceases once you turn off MPF, because what increased my rate of dynamic instability? Something getting phosphorylated by MPF that changed how my centrosome was working. All right? Okay, here's a picture of, what are the red things? Kinetochore proteins, and there's your kinetochore spindle fibers attached to them. All right, we've talked about tension. These are the three types of microtubules: asters, kinetochore microtubules. The book is calling them interpolar microtubules, but I like to call them what? Non-kinetochore. I think it makes more sense. They're both correct names, though. So there's two forces driving separation of the DNA. The non-kinetochore microtubules are elongating the cell. The asters are anchoring you to the cell membrane so you can have something to pull against. And the kinetochore microtubules are pulling on the DNA and separating the two strands. All right, here we have labeled in red is the kinetochore proteins. In green is the microtubules. And can you see where I have lots of microtubules? These are the kinetochore ones. They always end at a what? A kinetochore. And what's the blue stuff? Chromosomes, right? So we've used fluorescent labeling here. Here is a movie of cells dividing.
The mitotic spindle of the dividing human cell is reconstructed here in its full beauty from multiple optical sections that were recorded with a fluorescent microscope. Microtubules are stained in green, DNA is stained in blue, and the kinetochore, where microtubules attach to the DNA, are stained in pink.
Well,
How did they get it to, like, have that 3D image?
They took multiple slices. So I think it was a conglomeration of lots of pictures of the same kind of cell, so they're all the same size, and then they put them all together. Either that or they would have done, what's it called, confocal microscopy, where you have the cell and then you take a slice through where you're focused at this level, then you take another slice that's focused a couple microns lower down, and then the computer can put all those images together and turn it into a 3D image. All right, here's the same thing. We've stained the DNA, and now we're going to look at, in a syncytial fly embryo, remember in the first 13 divisions of a fertilized fly egg, the nucleus divides and separates. You do mitosis, but you don't do cytokinesis. You just get one giant cell with that in there. So here's a movie of this.
An early Drosophila embryo. In early Drosophila embryo, nuclei divide rapidly and in perfect synchrony. In this experiment, both DNA and tubulin are visualized with different fluorescent dyes. After the mitotic spindle is assembled, the microtubules, shown in green, start pulling the blue chromosomes to either pole. The chromosomes decondense and fill newly formed round nuclei. In preparation for the next round of mitosis, the centrosomes duplicate and migrate to opposite poles of each nucleus, where they form new mitotic spindles.
Okay. So see them just moving?
The whole embryo rhythmically contracts with each division.
There, I make a new mitotic spindle. I separate my DNA. Now I have one centrosome associated with each nucleus, and then it repeats. Here you're just looking at the DNA. Why are they all dividing at the exact same time and doing everything at the exact same time? Yeah, because they're all in the same cell exposed to the same MPF that's ultimately causing all this to happen. MPF being on starts it all, MPF being off ends it all. So pretty cool stuff. It'd be fun to be taking those first movies and figuring all this out, wouldn't it? Okay, so anaphase we've talked about. What are the two driving forces? Anaphase A is when your microtubules shorten. Anaphase B is when the spindles move apart as the cell elongates. So anaphase A is separating the DNA. Anaphase B is doing what? Elongating the cell. Which kind of fibers are these? Non-kinetochore. Which kind are these? Kinetochore microtubules. That's all they mean by anaphase A and B. They're both happening simultaneously, but it's different microtubules causing different effects. Here are securin, separase, APC, talking about that. So if what I drew here didn't make sense, take a look at this, and that'll help you understand that. So we've already gone over that. I'm not going to do it again. Here's a real picture of a cell in metaphase. Everybody's lined up. Now I'm in what? Anaphase. And there's anaphase A and B. So we've talked about all this. I'm just going to flip through. Now we're talking about cytokinesis. Is this a plant or an animal cell? Animal. How do you know? Yeah, it's forming a cleavage furrow. If you look really zoomed in close up, you can see how there's tension on the actual membrane. It's being pulled in because the actin has rearranged into a belt where your actin fibers are using myosin motor proteins to slide along each other and cinch that ring smaller and smaller and smaller. The actin is also attached to the membrane. I don't know if it's through focal adhesions or some other kind of attachment, and it's pulling the membrane in. Once the membrane gets pulled in enough, this bit confused with that, this bit confused with that, and I've now separated the two cells. So two kinds of cytoskeleton are involved. What are the green things? Microtubules, which formed the spindle apparatus, which did what?
Elongated the cell.
Separated the DNA and elongated the cell. And then this pink stuff is thousands of parallel, thick actin fibers, multiple actin strands lined up next to each other with proteins like alpha-actinin bundling them together. So you get strong, thick fibers, and then you have motor proteins, and that's causing them to cinch up, and that pinches your membrane in. All right, cells change shape a lot. Here is a cell during interphase. The cells are flat. They're adhered to the correct extracellular matrix, and you have all your organelles in their normal shape and structure. When you go into mitosis, do you see how totally different it looks like? You can't see any individual organelles. I'm taking up a lot less volume because I'm not flat and big. I'm round and small. Does that make sense? Now I'm finishing dividing, and as soon as I'm done doing that, I reestablish all my focal adhesions and my hemidesmosome junctions. That reconnects me to the matrix, and I go back to being flat again, and I get back to business as usual, or I'm back in what phase? Interphase again. Okay, so here's a dividing cell. All of this is M phase, and what does my cell look like? Is it flat or is it rounded? It's rounded. Now I'm finally finishing cytokinesis, and is my cell flattening out again? And now I'm back in my G phases again and ready to divide again. Okay, so lots of shape changes. It all involves the cytoskeleton.
You've given us the role of each. I mean, you've given us the key things with regards to the different MPF, APC. I mean, it's right there.
Exactly. Yep. These are the kind of things I expect you to know, but I want you to write more detail than that if you're writing it out. Yep. Exactly. Yep. So that's a good slide. How do plants do cytokinesis? They don't form a contractile actin ring. They do what? They form a new cell plate by lining up lots of vesicles full of cell wall material. How did these vesicles get formed? What delivery system? Yeah, the endomembrane delivery system. Okay, apoptosis. We've talked about apoptosis before, so I'm not going to go through that again. So we're done with this. So that was catching up from the last chapter. Why don't you go take a break, and then we'll start the new chapter.
Oh. This triggers telophase. APC triggers telophase.
How does it trigger telophase? It triggers anaphase by ubiquitinating what?
The
securin, which activates separase, which inactivates cohesins. It triggers telophase by phosphorylating who? The cyclin. Not phosphorylating, ubiquitinating M-cyclin. Then the proteasome destroys cyclin, and what happens to MPF?
Yeah.
It gets deactivated, and what's it no longer phosphorylating? It's no longer phosphorylating the lamins, and so eventually they become dephosphorylated, and when they do, they spontaneously reassemble into a nucleus.
Gotcha, okay.
Same thing with the condensins. I'm no longer phosphorylating them, so they're no longer sitting there inactive. They're now, or let's see, I phosphorylated to turn them on. When they get dephosphorylated, they get turned off, and they're no longer condensing my DNA, super condensed into chromosomes. Now I turn back into chromatin. So it's just because I turned MPF off at the M phase checkpoint that eventually telophase happens. Yep, this is telophase. Okay, great questions. So,
it's 2 intervals for into meiosis 1. We're referring to the fact that there's 2 types, so just because it's 2 divided
yep.
by 4.
And one way that it helps you know that there are 2 types is draw them a little different from each other. So maybe make this a fat chromosome, and this a skinny chromosome. Then it's easier to see that it's 2 types.
But we understand, though, because it's meiosis 1, it's not 4. I mean, there's 4.
There's 4 total
chunks.
chromosomes, but it's meiosis 1. So they lined up next to each other, and in anaphase, it's homologous chromosomes getting separated, not sister chromatids.
Okay, so then when you go here and you double it,
Yeah. So
And
so
that was what I
it's a
was
different
confused.
cell.
Yeah,
You're
so
doing
I
two
was
different
thinking
things. So this
eight,
one's
So I was like, there's got to be 8 little pieces in here, not 4.
exactly.
Okay, so I had a
So let's see. If I had 8 chromosomes, they would, and it's meiosis, they would have lined up
There would have been 4.
side by side.
Mm-hmm.
So like that. At the end of meiosis 1, what would I have in each cell?
That and that.
Yeah, I'd have 4 chromosomes. They look like they're homologs, but they're not. These are different, all 4 different chromosomes. All right? And then in, what did it say, meiosis 1 or meiosis 2?
Meiosis 2.
Yeah.
So then these
So
are
now
split. You
this
have
one
on each side.
will now line up like this. And so what are we in? We're in anaphase
Okay, so they're just
2.
barely
So
separating.
they'd be separated, and it'd be 4 different ones. But don't draw them like I did so they look like homologs. Make them so they're all 4 different from each other.
Would you please? Oh, sorry, Chris, turn it for me.
But you would have 8 sister chromatids
Okay, that makes
getting
sense.
separated from 4 chromosomes.
And then when we say that the difference between prophase 1 and prophase meiosis, we say there's. I guess
Hold on just a second. Did Skylar, I wasn't listening. What did you say? Okay. All right.
When prophase 1 different prophase and metaphase, mitosis, you've doubled the number of chromosomes because you're separating.
No, you start out
homologous
exactly the same. You've got replicated chromosomes. That's exactly the same. The difference is, and we haven't talked about it yet, in prophase of mitosis, the chromosomes condense wherever they want to be. In prophase of meiosis, when the chromosomes condense, they're also homologs are going to find each other. They're going to line up next to each other.
Saying is the difference. Is
That's
the
the difference.
homologs have lined
Yep.
up next to each
And
other.
then they're going to
the
do a
ones
process
separated.
called crossing over, and none of that happens in mitosis. But we haven't talked about that yet.
I was looking at it because I wrote that. I mean, I made the little picture. One of the things is like, there's 2 next to each other, and there's only 1.
Yeah.
As
So this,
one difference.
you've got 4 chromosomes
I mean,
there?
I did have 4 on each side,
Yeah.
but then I was looking at it,
Okay.
I was like, oh, wait, that's... And I was like, I should ask her, because it's like...
It's
this has
because
2 and
it's
equals 4.
because it's meiosis 2. In meiosis 1, I had 8 chromosomes. But at the end of meiosis 1, I've cut my number of chromosomes in half, so I only have 4 chromosomes now.
So would that be right then? Would it only be 4?
So in meiosis 2, yeah, you'd have 4 chromosomes, and the sister chromatids would be separating. So this is correct, except you need 4 chromosomes.
So you do need 4 on each side.
You need 4 on each side, because you have 4 different chromosomes. N is equal to 4 in this cell, and you still have 1 of every chromosome. Thank you. Okay, have any of you gotten emails from the college talking about course reviews?
Yes, course evaluation.
Did it say to go to my page to do them, or did it say to go to Canvas to do it?
It said to go to my page.
Okay.
SLCC, my SLCC.
All right, in 1610 they're changing it. I wasn't sure if they were changing it for all biology classes or just 1610. So yeah, if you guys would please do that, go on and do the course evaluation. It's under my page.
My SLCC.
So just, yeah, go to my SLCC, and it'll be a, yeah, up on the upper right. There should be a thing just to click on to do it.
I've already been on there telling the world how horrible 2020 is.
Those evaluations are totally anonymous. They won't affect your grade at all. I won't read them and say, oh, that was such a mean thing to say, and now I'm going to flunk them or something. Your grade is totally dependent on what your grades are in the class. I don't put any personal feelings into that. It won't help you at all, no. You can be as nice and schmoozy as you want, and it won't make a difference.
The amount of writing that we have done for you, you can tell who wrote what just by how we write.
That's probably true, but the fact is that...
And the truth shall set you free.
Yes, but anyways, the point is, it won't count anything towards your grade because you know me, it takes me weeks before I ever get to reading anything that you've actually written. And so it'll be long after your grades are posted that I will look at any of this. The point is, please fill those out and fill them out honestly because that is helpful to get feedback to improve the course. Of course, I appreciate positive comments, but it's perfectly okay to put negative comments on there too because that's how we learn and improve. If you fill it out, then you get an extra credit point.
Oh, there we go.
So the way you, yeah, one solitary, but they add up. So the way you get that is take a screenshot when you're done. It says, thank you for doing this, and it says what class it was. Just take a screenshot of that and email it to me, and you'll get your extra credit point. You can do that if you want, but no, I don't want that. Okay, now we need to move really fast because we took a lot of time doing the last chapter instead of the chapter we're on. So I'm going to go through these quickly because most of these things we've already mentioned. In bacteria, when they divide by binary fission, this is also reproduction because the cells are single-celled organisms. Is it sexual or asexual reproduction? It is asexual, meaning the daughter cells are genetically different from the parents or genetic clones. They are clones. Okay, so that's what it means by asexual reproduction. Sexual reproduction mixes up the DNA, and the offspring, the new cells, have a different arrangement of alleles, different combination of alleles. Lots of different, you don't have to be unicellular to reproduce asexually. There are some organisms, like the hydra, that can reproduce asexually also. The hydra is an ancient sea organism. It's really cool. It just has 2 layers of cells, an outer layer and an inner layer that does all the absorption and everything. It has an opening here where food can come in and where waste can go out. And when conditions are good, this hydra is surviving well. It says, I should have some kids. But does it want to make kids different from it? Or does it want to say, I want kids just like me because I'm perfect. I'm surviving really well in this environment. And so it reproduces how? Asexually, by cells here deciding to do mitosis or meiosis? Mitosis. So I get more diploid cells just like the parent. But you're also sending signals here, and these cells know they're not part of this organism, they're part of a new one, so they make all the cell parts. And once they're all done, I get a signal here to apoptose. These cells die, and now I have a little baby hydra that's an exact copy of the original parent, and all it needs to do now is grow bigger.
So in essence, if you're just cloning your cells, does that mean you're a normal?
Yes, technically, it does. Yep. So when is it beneficial to reproduce asexually?
When there's not other mates around.
Okay, that is one thing. If you can't find a mate, it'd be really nice if you could just reproduce asexually. That would be awesome. Think about, there's no need for genetic diversity. Why wouldn't there be a need? Because the environment is stable or because it's unstable and changing?
Stable.
Yeah, so in a stable environment, then things tend to reproduce asexually. When the environment changes, then it's advantageous to reproduce sexually. Why?
Because the offspring are going to be different from the parents.
Yeah, they're going to have slight differences, and hopefully at least 2 of them, preferably a male and a female, will have different enough characteristics that they can survive in this new environment and keep the population going. Okay? So what's the advantage of sexual reproduction? It gives you what in your population? Diversity or variation so that you can do what in changing environments? Evolution or adapt to changing environments. And even though some organisms won't survive, the species will survive. All right, here we see a picture of this. When you look out at the trees in the fall, and go out and look at the gorgeous aspens, do you notice that there's clumps where they all are sort of the same color? Like here's much more yellow ones, here's more orange ones, here's super orange ones. Why are they all occurring in a clump? Because trees are able to reproduce, or at least these kinds of trees. Yeah, they send out rhizomes. So here's my beautiful tree that I've planted in my yard and that I want there. But what do aspen trees do? They grow their roots out this way and periodically send a sprout upwards. And when it gets above the grass level and the sun hits it, signaling happens that causes it to become leaves and stem and not root anymore. Or I get a baby tree. What's causing all these cells to be formed? Are we dividing by mitosis or meiosis? Mitosis, or in other words, are all these cells exact clones of this original tree? And so why do I get clumps of color where they're all exactly the same? Because technically they're all the exact same tree. And interestingly, the largest organism on Earth is Pando, and it's here in Utah, up in the Uinta Mountains. It's about 106 acres of aspen trees, but they're all clones of the same tree.
When they go to leaf, they all go to leaf at the same time.
Yeah. So, interesting stuff. Okay, so now we're going to look at sexual reproduction. So here we have two, whatever these things are. I didn't watch Sesame Street when I was young, so what are these things called? Fraggles. Fraggles. So, two fraggles, and they each produce gametes. The gametes come together to make a zygote, and then the zygote divides and divides and divides to make new offspring, which are similar to the parents,
Did the Muppets really do a show?
but not identical. Okay, sorry that this color is wrong. When I copied this slide, it went black instead of white. But we're now asking, are we haploid or diploid? This is the parent generation. Are they haploid or diploid? Yeah, most of the cells are diploid, but the gonads of the male, which is testicles, are making haploid what? Gametes called sperm, and those are haploid. The female, her gonads are ovaries, and they are making haploid eggs instead. All right? An egg and a sperm comes together to make a zygote, which is the first cell of the next generation. It's back to being diploid again. What did you say, Chris?
Nothing.
Okay.
Did the Muppets really do a show on this? I didn't realize that they...
Not that I know of, no. We just took pictures of Muppets and turned them into this.
I was about to say,
Okay.
well, ruined my childhood right here.
All right, and then you end up with your F1 generation or the offspring. So the question is, we're going from being diploid to being haploid back to being diploid again in the next generation. What are the processes that allow us to do that? How do you form a zygote? What's the process called?
Two haploids from the parents come together and
What's the process?
fertilization.
Fertilization, exactly. Okay, why don't I have it written there? Somewhere I have it written.
It's the third one.
Okay.
Keep looking at your slide there.
Anyways, so this is fertilization. What kind? Oh, that's because I didn't ask it yet. How do the cells divide to go from a single cell to multi-celled organisms? I want more cells so I can grow bigger. This is growth and repair, or I'm dividing by what process? Mitosis. I take a diploid zygote and make a multicellular diploid organism. All right? How do I make gametes? By meiosis. The purpose of meiosis is to cut your ploidy in half, go from two end to one end. And then fertilization is the process of uniting the two gametes to make a zygote. All right? So that is cell cycles. Here is an egg. Eggs are usually large because they need to be packed full of lots of building materials. Sperm are usually tiny because all they're donating is the genetic information and the trigger to get things started. And then the nucleus of the sperm and the nucleus of the egg fuse into a new diploid nucleus that has two sets of chromosomes. We've talked about this before. Remember, we talked about the calcium wave moving across the egg when fertilization occurs. What does this calcium wave do? First, it triggers vesicles to fuse with the membrane. Is this regulated or the constitutive pathway, endomembrane vesicle pathway? Is this regulated or constitutive exocytosis? What did you say? Regulated, because they're not fusing until a sperm hits and we get this calcium signal, and then I fuse. So that's an example of that. Here's a movie on this.
Sea urchin egg during fertilization is visualized here simultaneously by phase contrast microscopy and by fluorescence microscopy. The egg contains a fluorescent dye that becomes brighter in the presence of calcium ions. When a sperm cell fuses with the egg, the fluorescence image shows a wave of calcium ions that sweeps through the cytosol, starting from the initial point of sperm-egg fusion. Following the path of the calcium wave, we see a membrane called the fertilization envelope rising from the
See how it lifts right off there?
The fertilization envelope protects the fertilized egg from the outside environment and prevents the entry of additional sperm. The rise in cytosolic calcium triggers an elevation of the fertilization